introduction material so we know exactly what we're talking about. And like Dr. Cabot said, the majority of my talk will focus on Chapter 2, looking at GRP 94 and aggregation prevention. And I will briefly touch on some data and conclusions for Chapter 3, followed by a summary of my work presented here. Before I can tell you about GRP 94, I should tell you about molecular chaperones, what they are, and why we need them. In their infancy, proteins are going to be synthesized as polypeptide chains that will emerge from the ribosome, and they're going to emerge as unfolded or misfolded proteins. And as we know, for a protein to function correctly, it needs to be into the correct shape or conformation, otherwise known as its native state. There are chaperones associated with the ribosome that will capture these chains and facilitate their proper folding. However, a large majority of these polypeptide chains will emerge in the absence of a chaperone for subsequent identification by its chaperone for promotion of proper folding to the native state. In the absence of molecular chaperones, misfolded or unfolded proteins can form non-native contacts with each other, subsequently forming protein aggregates. And in addition to protein aggregation, unfolded or misfolded proteins can be targeted for degradation. My talk is going to focus mainly on the prevention or protection from aggregation aspect of molecular chaperones. And the reason we're so invested in researching protein aggregation is it's a major driving force for severe neurodegenerative diseases that we're probably all familiar with. Diseases like Alzheimer's, Parkinson's, and Huntington's disease are all a direct consequence of protein aggregation. And this is the reason we study molecular chaperones, to further elucidate how protein aggregation, prevention of protein aggregation can provide answers for solutions to protein misfolding diseases. Speaking of molecular chaperones, the HSP or heat shock protein molecular chaperones are a large group of conserved proteins. There are many different families, however, I will focus on HSP70s and HSP90s. HSP70s and HSP90s are present in 3 cytosolic eukaryotic compartments, where we have HSP70 and HSP90 in the cytosol, mitochondrial HSP70 and TRAP1 in the mitochondria, and of particular interest to us, BIP and GRP94 in the endoplasmic reticulum. Also important to note, HSP70s and HSP90s work with cofactors. HSP40 co-chaperones, which in the ER are ERDJ proteins, and the nucleotide exchange factor, which in the ER is GRP170. Introducing GRP94, the resident HSP90 of the ER. GRP94 assembles as a homodimer and shares conserved domain organization and approximately 50% sequence homology with its cytosolic counterpart. We see first an N-terminal, or the N domain, where we see nucleotide, peptide, and calcium binding, followed by a middle, or M domain, where we see nucleotide hydrolysis and potentially client binding, followed by the C domain, which is our dimerization interface, and where we also see client binding in GRP94. GRP94 undergoes conformational cycling as a result of nucleotide binding and hydrolysis, where we start with GRP94 in the open or apo conformation in the absence of nucleotide. Upon nucleotide binding, we form a closed conformation where the 2 protomers are twisting amongst themselves. And an ATP hydrolysis event is going to relax that structure, and subsequent release of ADP is going to reset the cycle and open GRP94 back up to the open conformation. Looking further into GRP94 function, GRP94 does function outside of its role as a molecular chaperone, working on the cell surface in immunity and peptide binding. So just briefly, GRP94 is required for lymphocyte development and activation, which subsequently means that it regulates cytokine production and activates immune stress responses. In line with that, GRP94 in general is going to regulate cellular homeostasis via these immune pathways. Similarly, GRP94 has been shown to bind peptides, where presenting peptides to T cells promotes the maturation of antigen-presenting cells. Looking at our schematic of GRP 94 activity, we can see that there are different requirements for different activities. So for the immune functions, we see that ATP binding and hydrolysis are required, whereas calcium binding is not. And the full length of the protein has been characterized in these activities. In comparison to peptide binding, which is independent of ATP binding and hydrolysis, but is regulated by calcium binding, and thus far has been identified at the N and C terminal domains. Now investigating the chaperone function of GRP94. GRP94, of course, functions in protein folding, both assisting in the folding of newly synthesized immunoglobulin chains, and preferentially folding and assembling intermediate and late-stage clients like TLRs, IGFs, and integrins. We also know that GRP94 collaborates with other ER chaperones in vitro remodeling assays that remodel denatured proteins. So protein folding activity looks very similar to its immune activity, where ATP binding and hydrolysis are required, where calcium is not, and the full length of the protein again has been characterized in protein folding. Last but not least, we look at aggregation prevention by GRP94, which, as we can tell by the question marks, is significantly underrepresented in research. There is a report of the MC domain preventing aggregation of a client mycilin, whereas the N-terminal domain was found to accelerate aggregation of that client. Similarly, another paper identified the C-terminal domain could prevent aggregation of protein kinase CK2, but could not prevent aggregation of malate dehydrogenase. So as of now, there is little known about the requirement for ATP and calcium in GRP94 aggregation prevention, which led to the development of my research questions, looking at, are there other client binding regions in GRP94? What are the requirements for aggregation prevention? And does GRP94 sample client-specific chaperone mechanisms? Which leads us directly into chapter 2, where I will spend the majority of my time looking at the N-terminal domain in GRP94 chaperone activity. In order to assess the ability of GRP94 to prevent aggregation, we first identified a model client, citrate synthase. Citrate synthase aggregation pathway is well characterized, and we see a distinct pattern of aggregation at increasing temperatures, suggesting that this is an appropriate client for our aggregation prevention assays. So we carried out our fixed angle light scattering assays to monitor the amount of light scattered, or the amount of aggregation of citrate synthase. So native citrate synthase was heat denatured at 45 degrees Celsius, either in the absence or presence of GRP94, and the subsequent light scattering signal was measured. So we can see this first line in black corresponds to citrate synthase at a concentration of 0.25 micromolar alone, and we observe a dramatic increase in the light scattering signal, which indicates we are observing citrate synthase aggregation. As we work our way through the graph, we can see as we add increasing concentrations of GRP94, we are significantly reducing the light scattering signal, which indicates a prevention of aggregation by GRP94. As a control, we also show that the highest chaperone concentration tested does not exhibit any aggregation properties and therefore is not contributing to the signal. Moving forward, we took the average of the time points between 1500 and 1800 seconds on our time trace graph to create the bar graph that represents the average aggregation seen at each concentration of GRP94 compared to citrate synthase alone. So looking here, we have our black bar, which again is citrate synthase alone, compared to the average aggregation at each GRP94 concentration tested. And we can see that as we increase the concentration, we are in fact decreasing citrate synthase aggregation. We also utilized statistical analysis to compare the aggregation seen at each concentration to our client alone. And we determined that at the 3 highest chaperone concentrations, GRP94 was able to prevent the thermal aggregation of citrate synthase to the highest degree of significance. We wanted to further characterize the aggregates, the citrate synthase aggregates, so we turned to dynamic light scattering, or DLS, where fixed angle light scattering tells us how much aggregation. DLS is going to tell us how large those aggregates in solution actually are. So if we look first at native citrate synthase, we can see it's quite small, around 1 nanometer give or take. And citrate synthase, heat denatured in the absence of a chaperone, forms very large aggregates, centered around approximately 350 millimeters. As controls, we show that native and heat-treated GRP94 are relatively similar to one another, indicating that heat treatment does not affect GRP94. And we see that when citrate synthase is heat denatured in the presence of GRP94, that large aggregate population is shifted to the size of GRP94 alone, confirming our fixed angle light scattering data that shows a prevention of aggregation by GRP94. We further questioned whether GRP94 would be able to prevent the aggregation of chemically denatured citrate synthase, similar to that of thermally denatured citrate synthase. And so what we found was with chemically denatured citrate synthase, GRP94 was able to prevent its aggregation to a similar degree as the thermally denatured citrate synthase. However, DLS shows that when GRP94 is in reaction with chemically denatured citrate synthase, the large population of aggregates is not completely resolved like we saw in thermal denaturation. Therefore, continuing forward, I will be presenting thermally denatured citrate synthase data. Given GRP94's role in binding ATP and calcium, we question the importance of ATP and calcium in aggregation prevention. What we see here is a composite graph that compares aggregation prevention by wild type, GRP94 with no additives in black, followed by GRP94 additive conditions. So we have GRP94 and ATP in stripes, GRP94 and calcium in checkers, and lastly we have the combination of GRP94 with ATP and calcium in the light bar. What we found was that at each concentration tested for GRP94, there was no significant difference between any condition tested with wild-type GRP94. The lack of significant differences in aggregation prevention suggests that ATP and calcium are not required for aggregation prevention by GRP94. This was peculiar given that ATP binding and hydrolysis are required for GRP94's protein folding function. So we further probed the specific activities of ATP binding and ATP hydrolysis in aggregation prevention. We utilized an ATP binding mutant, D149N, that cannot bind ATP, and an ATP hydrolysis mutant, E103A, that can bind but not hydrolyze ATP. In our fixed angle light scattering experiments, we determined that GRP94 D149N was not significantly different at any concentration tested when compared to wild-type GRP94 or wild-type with ATP. We saw a similar trend with E103A, where again, at all concentrations tested, there was no significant difference in its ability to prevent citrate synthase aggregation. DLS confirms that with both D149N and E103A, we have a loss of the large citrate synthase aggregates, and that population is shifted to the size of GRP94 alone. Again, confirming that at our highest chaperone to client ratio, we are effectively preventing the formation of large citrate synthase aggregates. So together, this data tells us that neither ATP binding nor hydrolysis are required for aggregation prevention by GRP94. Published work by Dr. Yamamoto from our lab investigated the role of nucleotide in GRP94's conformational states using Deer spectroscopy. So for this experiment, she created a cisless GRP94 construct, meaning that all of the native cysteine residues were mutated to alanines, so that she could intentionally incorporate a cysteine residue for site-directed spin labeling. Residue K161 was identified based on crystal structures of GRP94 that show difference in distances between residues on opposing protomers in different GRP94 conformations, meaning that in a closed conformation, residue K161 on opposing protomers will be closer together compared to residues on opposing protomers in the partially open structure. Using an in silico simulation with a labeled GRP94 at position K161, predictions confirm that we are able to distinguish between closed and partially open conformations, suggesting that K161 is an efficient site for determining differences in GRP94 conformational states in response to nucleotide. And so what she found was that GRP94 in the apo conformation, or in the absence of nucleotide, GRP94 populates a largely open population with distances larger than predicted, suggesting that we're actually sampling a more open conformation. Similarly, in the presence of ADP or ATP, that more open conformation is still observed, suggesting that nucleotide doesn't really have an effect on the conformational equilibrium of GRP94. She also identified that incubation with AMP-PMP, which is a non-hydrolyzable ATP analog, this could trap GRP94 in a closed conformation, where we see the presence of a large population with distances that correspond to those predicted for the closed conformation. So as of now, we have only been probing the open conformations of GRP94, as our conditions have been in the absence or presence of ATP. And so we question whether the closed conformation of GRP94 would have negative impacts on aggregation prevention, considering that the previously identified C-terminal region would be unavailable for client binding. To that end, we incubated GRP94 with AMP-PMP and utilized this in our light scattering assays. And what we found was that GRP94 in a closed conformation prevents aggregation of citrate synthase similarly to that of wild-type GRP94, and DLS again confirms the complete reduction of the size of citrate synthase aggregates, confirming that GRP94 in a closed conformation can also prevent aggregation of citrate synthase, so the global conformation of GRP94 is not relevant for aggregation prevention. So thus far, we have determined that GRP94 does not require ATP or calcium binding, and in addition, all solution forms can prevent aggregation. So if you will join me in observing our question marks turning into X's, oohs and ahs are appropriate. So moving forward, we questioned what region of GRP94 was responsible for preventing citrate synthase aggregation, given that previous reports identified mainly the C-terminal domain in aggregation prevention. Interestingly, we found that only the N-terminal domain was responsible for preventing aggregation of citrate synthase, with a pattern similar to that of wild-type GRP94, where we see a decrease in aggregation as we increase the concentration of our domain. DLS, again, you guessed it, confirms that the large citrate synthase aggregates are reduced to the size of the domain alone. In contrast, we see that the M and the C domains have absolutely no effect on the aggregation prevention of citrate synthase. And in fact, new DLS for you, we can see that in reaction with the M and the C domains, that citrate synthase aggregates persist. We questioned whether the N-terminal interaction with citrate synthase was specific for citrate synthase, or if this could be more globally applied. So we tested another model in vitro substrate, luciferase. And what we found was, in addition to full-length GRP94 preventing luciferase aggregation, only the N domain could prevent aggregation of denatured luciferase, with no prevention being observed from either the M or the C domains. Looking at DLS, we can see that a large population of luciferase aggregates, either in the presence of full-length or the N-terminal domain of GRP94, show a shift in the size of those aggregates, which confirms the aggregation prevention we see in our light scattering assays. Similarly, in reaction with the M or the C domains, the large population of citrate synthase aggregates remains. The role of the N-terminal domain in aggregation prevention was interesting to us, especially considering that the N-terminal domain also harbors a peptide binding site. So we questioned whether this peptide binding site in the N-terminal domain was participating in the aggregation prevention that we observed in the N-terminal domain. A previous group identified residue H146 as the GRP94 peptide binding site, and mutation to H1460 abolished peptide binding. So to test whether this region was participating in aggregation prevention, we used this mutant in our light scattering assays. In our light scattering assays, we used GRP94 at both limiting and saturating chaperone concentrations, meaning that at limiting chaperone concentrations, there is less GRP94 than client present, versus saturating conditions where there is more GRP94 present than client. So what we found was that GRP94 H1460 was significantly better at preventing citrate synthase aggregation at limiting chaperone concentrations. We observed a similar trend for luciferase aggregation prevention, where again, at these limiting chaperone concentrations, H1460 is well equipped to prevent aggregation when compared to wild type. DLS, to follow suit, confirms that both the citrate synthase and luciferase aggregates are returned to the size of H146D alone, confirming the aggregation prevention we see in our fixed angle light scattering. This observation was peculiar given that H1460 was shown to abolish peptide binding. So we questioned whether the increase in aggregation prevention would translate to an increase in client reactivation. So to test this, we used a luciferase reactivation assay that couples aggregation prevention by GRP94 with protein folding by another ER chaperone system, the BIP system. Introducing more players. BIP, the HSP70 in the ER, in its open or ATP bound conformation has low affinity for clients. However, a J protein co-chaperone is going to deliver the misfolded client to BIP, and the direct interaction between the J protein co-chaperone and BIP is going to facilitate an ATP hydrolysis event. Following that hydrolysis event, the J protein will dissociate, and a conformational change is triggered where the substrate binding lid traps the misfolded client in the substrate binding domain of BIP. Subsequent nucleotide exchange replaces the hydrolyzed ATP with a fresh ATP, releasing the folded client and opening BIP up for another cycle of client binding and release. Published work from our lab shows that luciferase, when heat denatured in the presence of GRP94 and ATP, can then be released into solution by GRP94 for further refolding by the BIP system. This was the perfect assay to test our question about whether H1460 would increase client reactivation. So in our assay, we found that when luciferase was heat denatured with wild type GRP94, GRP94 could release luciferase into solution, and BIP could refold approximately 15% of the native luciferase activity. However, when luciferase is denatured in the presence of H1460, we observe an inhibitory effect where we only see approximately 6% reactivation. This suggests that GRP94 H1460 is interacting stronger with its clients, preventing its release into solution for further refolding, indicative of the decrease in reactivation that we see. Adding to our conclusions, we have now identified the N-terminal domain of GRP94 in aggregation prevention for the first time. And we've also identified or determined that a peptide binding site is participating in this aggregation prevention. So if I can draw your attention to our aggregation prevention regions, we can now add the N domain. Okay. So further looking at the H1460 mutation, we see that there is an increase in aggregation prevention, and we question whether the change of this charge was increasing electrostatic interactions with client proteins. In line with that observation, a previous group identified that the N-terminal domain accelerated aggregation of myocillin, whereas the MC domain prevented aggregation. Further investigating this aberrant interaction with the N-terminal domain, this group identified crosslinks with two lysine residues in both the pre-N and N domain of GRP94, and they determined that truncation of this pre-N domain rescued the increase in aggregation that was observed with the N-terminal domain, suggesting that the pre-N domain was important for this client interaction. So given the proximity of these residues with 146, we questioned whether the pre-N domain would be required for aggregation prevention of our clients, citrate synthase and luciferase. To test this, we used a delta 72 GRP94 construct where the pre-N domain has been truncated. And what we find is that in light scattering assays with thermally denatured citrate synthase, GRP94 delta 72 prevents aggregation to a similar degree as wild type, suggesting that the pre-N domain is not required for aggregation prevention of citrate synthase. However, when you look at luciferase, at saturating chaperone concentrations, GRP94 delta 72 is significantly impaired in its ability to prevent luciferase aggregation, which suggests that the pre-N domain is required for interactions with luciferase. We have VLS that confirms the resolution of citrate synthase aggregates in the presence of GRP94 delta 72. However, with luciferase, we see that the large aggregates are not completely resolved. They are shifted to a smaller size. However, confirming our light scattering data, aggregation is not completely null. There is still aggregation occurring. So we've also concluded that the GRP94 pre-N domain is required for aggregation prevention of some clients, but not others, suggesting client-specific chaperone mechanisms and perhaps distinct client binding regions along the length of the protein. Future directions for this project should include investigating the increase in aggregation prevention seen with the 146D mutation, investigating electrostatic interactions of peptides with GRP94 to determine if these are the driving force behind client-chaperone interactions. And lastly, determine what regions of GRP94 interact with biologically relevant clients, like TTR and VSDL, to determine additional client interaction services on GRP94. With that, as promised, I will briefly touch on Chapter 3, looking at J proteins, ERDJ4, 5, and 6. So there are 8 J proteins in the endoplasmic reticulum, where ERDJ1 and 2, and 7 and 8 are membrane bound, and ERDJ3 through 6 are proposed to be luminal chaperones. While it is important to note that J proteins differ greatly in their structure and domain organization, they all share a highly conserved J domain, hence the name J protein. The J domain is what is responsible for direct interactions with BIP that facilitate the ATP hydrolysis cycle we saw previously. More specifically, an HPD motif within the J domain is responsible for that interaction, and mutation to QPD is going to inhibit that interaction with BIP. While all J proteins are not created equal, they all are created with one purpose, and that is to work with BIP in either a pro-folding or pro-degradation capacity. So ERDJ1 and 2 are membrane proteins that have been characterized in pro-folding and assisting the import of nascent polypeptide chains and their subsequent folding. ERDJ3 has also been characterized in its pro-folding role, collaborating with ER chaperones in protein remodeling. When I started here at Miami, my original goal, as you heard, was to further characterize ERDJ4, 5, and 6 and their abilities to collaborate with BIP and the ER chaperone system. However, I learned through lots of trial and error and lots of trash that ERDJ4 and 5 were not for me to purify. It wasn't in my cards, and that's okay. So this slide is going to serve a couple of purposes. One, it's to spare my audience the many images of gels that never produced any protein. And it's also to provide a short, comprehensive list of things of what not to do to my current and future grad student friends, Io and Chioma. If you ever decide to pick these up, this is a non-comprehensive list of things of what not to do. If you want more details, please see my dissertation. A couple of things. Please don't do cation exchange chromatography. Your protein will not bind, and if it does bind, it's going to be insoluble if you try to do something with it. Similarly, if you try to dialyze your protein out of urea, it's going to precipitate. So actually a better idea is to not use urea at all. And the last one I want to touch on is changing the growing protocol, changing the growing and induction temperatures, changing IPTG concentrations. It doesn't work. It doesn't increase solubility, and it doesn't increase expression. I've done that work for you. You can check it off. So try something else. While ERDJ4 and 5 might be unsuccessful for now, I do have a success story to share with you in ERDJ6. ERDJ6 was the first protein I ever successfully purified here in Miami, so this was a big deal. So after lysis and centrifugation, the supernatant fraction was applied to his trap affinity chromatography, and we can see a beautiful ERDJ6 band where these indicate fractions of protein that were eluted from the column. And so all of this beautiful protein was pooled together and dialyzed overnight to prepare for anion exchange chromatography, where we see further purification or cleaner protein, if you will, where a couple of fractions, 2 to 3 fractions from anion exchange, were pooled together and applied to a size exclusion column for the final step of purification, obtaining purified ERDJ6 that was further characterized in preliminary assays with BIP. Given the role of J proteins in their interactions with BIP, which I've touched on a couple of times, it was important to determine if our ERDJ6 protein was able to stimulate BIP ATP hydrolysis activity. We found that although ERDJ6 has some background activity alone, that at saturating concentrations, ERDJ6 can adequately enhance the ATP hydrolysis rate of BIP, which is very low on its own. Forgot to mention that we did this using an enzyme coupled assay where we use pyruvate kinase and lactate dehydrogenase enzyme mechanisms, and ultimately we monitor the oxidation of NADH, which directly correlates to BIP's ATP hydrolysis activity. I also generated the QPD mutant of ERDJ6 that should be catalytically inactive. And in fact, I did find that the QPD mutant was not able to stimulate BIP's hydrolysis activity. We also made an interesting observation looking at the ERDJ6 size exclusion elution profile. So we first have this kind of what I've referred to as a lagging peak, or a large volume of species that are larger eluting before our expected ERDJ6. This pattern is similar to those seen with proteins that form higher order oligomers, so we question whether that was happening with ERDJ6. In fact, a preliminary native page analysis shows that we are forming oligomers, but to what extent we're not sure yet. That is in the books. So an ERDJ6 sample from the beginning of the elution profile should correspond to a large oligomer or a large species, and that is in fact what we see, where the sample really didn't migrate through the gel. It's very large and it wasn't able to work its way down. Compared to a sample of protein from the end of the elution profile, which should show smaller species, and in fact that's what we see, as there is some migration, and the distinct presence of at least 2 species that should be further identified. So basically what I'm trying to tell you in this whole chapter is that ERDJ4, 5, and 6, after they're purified and characterized, should be implemented into the ER quality control system to determine their interactions with the chaperones. So do they work like their brother protein, ERDJ3, ERDJ3 collaborates in protein refolding and actually enhances client transfer between BIP and GRP94. In ERDJ4, 5, and 6 function in this capacity in protein refolding, or are they destined for pro-degradation roles with BIP in clients? So ultimately, new purification protocols need developed for ERDJ4 and 5, starting at the top, using cells that are designed for membrane protein expression. And then ultimately, using all ERDJ4, 5, and 6 in ATPase and luciferase assays to evaluate their ability to collaborate with the chaperone system. So as a summary of my work here, in chapter 2, the data presented is going to expand the current knowledge of GRP94 aggregation prevention. We elucidated requirements, or a lack thereof, for aggregation prevention, and determined the participation of a peptide binding site. All of this information together paves the road in front for future researchers to identify additional client recognition surfaces in GRP94, advancing the field in chaperone client recognition. And ultimately, we can investigate how toxic protein aggregates are escaping the quality control system in hopes of developing medical interventions that can prevent the onset or progression of serious disease. And while chapter 3 did not yield the world's best successes, I did actually unveil some interesting information regarding the identity of ERDJ4, 5, and 6 that can provide clues for their actual function in the cell. So for ERDJ4 and 5, if they are in fact associated with the membrane and are participating in a pro-degradation capacity, identifying clients that depend on ERDJ4 and 5 for export opens doors to exploit this function and hopefully develop, again, interventions that can prevent these diseases caused by protein aggregation. And similarly with ERDJ6, we observe that it's forming oligomers, and this could be interesting in the dual functionality of ERDJ6. It could be possible that one oligomeric species is working in protein remodeling, where another species is working in pro-degradation. And so ultimately, everything I have provided you here today falls under one big picture, and that is, how are molecular chaperones recognizing their clients? How are they preventing their aggregation and their degradation? And how can this be mitigated to resolve diseases that affect people that I personally know and love? So with that, I would like to thank everybody here today, especially my committee, and especially Dr. Kravitz. Thank you for your support and all of your help over my time here. I have to give a shout out to all of my past and current lab members, Dr. Ya Mangwa, she's my rock. All of my other friends, Io, Chioma, Hannah, IK. I couldn't have done this without you guys. You're fantastic. I had incredible undergraduate students. Shout out Kylie. To my family, I couldn't have done this without you. My husband, I appreciate everything. And to my friends, Allison, you know. So with that, thank you.